Good morning, everybody. Can you hear me well? Yes? Okay. Before anything else, I want to express all my gratitude to Dr. Christian Fiala and to Dr. Otta Koenig for their hospitality, for their most efficient organization of this conference, in which I'm extremely pleased to participate with you this morning. As a matter of fact, I don't feel I'm going to take all my time because perhaps a little bit by contrast with other speakers we have had a chance to listen to for now more than a day, I'm not going to review a lot of background on all the shortcomings of the HIV/AIDS hypothesis. These have been magnificently reviewed and analyzed by several of our speakers, and I'm not going to go into that. As a matter of fact, I would like to restrict my presentation this morning to one single point, which is really summarized in the title of my presentation. I would like to attempt to convince you that to properly analyze AIDS research, we have to remember the existence of human endogenous retroviruses, which are interfering heavily in the interpretation of AIDS research for the past 10, 20 years. And since I'm going to use the words endogenous and exogenous quite frequently in this presentation, maybe I want to make crystal clear that you have no problem with these two words because they are keys to what I'm going to attempt to convince you about. Endogenous viruses are not that many, but the best known are unquestionably the human endogenous retroviruses. And if you want to convince you about the importance of the current literature on human endogenous retrovirus, HERV, just make a visit to Google and open human endogenous retroviruses, and you'll spend hours to try to survey the importance of the literature on these viruses. Endogenous meaning integrated in the human genome, therefore transmitted vertically in a Mendelian manner. But endogenous retroviruses are perhaps not the only viruses that correspond to this characteristic. I learned kind of recently about Borna virus that might be another case of genome integrated, therefore properly endogenous viruses. By contrast, tell me, I hear there is such a resonance here, maybe, no, I cannot hear you better like that. I need this because I have a severe hearing problem and, no, it's better like that, sorry. Exogenous viruses are, of course, infectious viruses responsible for contagious disease and for epidemics. So these are completely two separate words. HIV has been regarded as an exogenous infectious virus and has therefore nothing to do with HERV, with the human endogenous retrovirus. These are two completely separate words, but all the observations made on HERV have been interfering very heavily on the analysis and the interpretation of HIV/AIDS research, and that's the point I would like to try to convince you about this morning. It's true to say that scientifically, HIV has never been scientifically proven to exist. So to say that HIV does not exist is not a very scientifically appropriate statement, basically because it's extremely difficult scientifically to prove that something does not exist. So if you prefer to say, which is more cautious wording, that HIV has never been proven to exist, as has been claimed for more than 20 years by now, that's perfectly all right with me. But HIV is not an infectious agent, and perhaps the most convincing facts confirming this point are, number one, that AIDS is not observed among prostitutes, and if HIV would really be sexually transmitted in the heterosexual population, we would, of course, expect prostitutes to be very high on the risk group of AIDS, and they are not. And this fact has been recognized for a long time. If I well remember, it was Ruth Bernstein in 1993 who started to speak about the prostitute paradox, claiming that indeed we do not find AIDS among prostitutes, and those who are found HIV positive are always those who are also intravenous drug users. Another fact that since the very beginning of the entire HIV/AIDS history is indicating that there is something very suspicious about the official hypothesis is the fact that for the first many years of the so-called AIDS epidemic, all victims, or practically 95% or plus of the victims, were of the male gender, and I never heard about an infectious disease which has such a gender-specific characteristic. Another problem is that HIV has never been isolated nor purified directly from a single AIDS patient. All claims made very repeatedly in the literature for successful, quote, quote, isolations are based on the recognition of so-called molecular markers. Molecular markers that have been mostly identified in complex cell culture systems, if not directly in AIDS patients. But the specificity of these so-called molecular markers has never been verified because the total lack of success of all attempts to purify, to isolate and purify the HIV virus. That statement has been made for many years, and I guess was initially made by the Perth group back in the 1993. So without successful isolation and purification of HIV, the specificity of molecular markers has to be put under considerable large questioning. But more precisely to our subject, if we make the simple claim that, quote, quote, HIV does not exist, it's not scientifically acceptable because it's not going to give us any position to respond to three questions, which are basically misleading factors listed on the screen now. If, quote, quote, HIV does not exist, how do we explain viral load? What are we talking about when we keep speaking and analyzing in all the AIDS patient viral load? If HIV does not exist, we have to answer that. The number two confusing question has to do with the extremely numerous claims made to the fact that HIV never stops mutating, and the frequency of these mutations has been emphasized by many as an explanation for the lack of success in all the attempts to prepare HIV vaccine. A third misleading factor that also gave an apparent credibility to the entire HIV hypothesis is the fact that in the historic paper published in 1983 at the Pasteur Institute in Paris, an excellent electron microscope picture was published representing most typical retroviruses budding on the surface of some cells. So these are the three factors that cannot be faced and answered properly unless we look for an alternative explanation for the analysis of the entire HIV/AIDS dogma. We need clarification for these three factors. And as I hope to convince you this morning, these three factors can all be very reasonably and scientifically explained by the presence and the interference of human endogenous retroviruses. We all carry human endogenous retrovirus in our chromosomes. You, me, and all the others. It's universal. My attention to that problem was first attracted by a most significant paper published, I guess, in 1996 in Berlin by Löwer. And the paper was published in the PNAS, and the title included the words, "Viruses in all of us." And it's true. It's not a hypothesis. It's a fact that cannot be ignored. They are, as we said, vertically transmitted. They are probably almost always defective viruses, not producing recognizable particles that one could see under the electron microscope. But there are exceptions to that in the cases of some embryonic cells, where HERVs can express themselves by the formation of particles that can be readily recognized under the electron microscope. And the most classic case of what I just said is the human placenta, in which typical retroviruses, which are of endogenous origin, can be very clearly recognized, as demonstrated many years ago by several authors. Reference is not appropriate here. HERV are of questionable pathogenicity, but there are a considerable number of people who are studying the possible, the hypothetical pathogenicity of HERV in the direction of cancer and of some autoimmune diseases. But what's more significant is that when the analysis of the human genome was terminated a couple of years ago, it became quite evident that approximately 8% of our genome is represented by typical nucleotide sequences that are of retroviral nature. So that's the key point you have to remember, because it implies that any single molecule of our DNA, wherever that molecule originates from, contains about 8% of nucleotide sequences which are extremely similar, if not identical, to the retroviral genome. Another extremely important fact that became clear a little bit more than 10 years ago is that we all carry in our peripheral blood circulating DNA. This was most clearly demonstrated by a nucleic acid biochemist from Geneva, Anker, I guess in 1999. And it's in all of us in variable amounts. And the amounts of circulating DNA in our peripheral blood can fluctuate in accordance with our various pathological conditions, or perhaps also caused by stress, caused by infection, or caused by apoptosis. All these factors can determine an increase in the amount of circulating DNA in the blood of healthy, but perhaps more convincingly of people affected by a variety of pathological conditions. Now, with these 2 points well in mind, one, that any human DNA includes 8% of retroviral sequences, two, that we all carry circulating DNA in our peripheral blood, we turn to the techniques used for determining what's currently referred to as the viral load. Viral load seems to imply that there is viremia. The viremia meaning the presence of viral particles recognizable under the electron microscope and present in the peripheral blood. Here we hit a major problem. Nobody has ever demonstrated one single particle of retrovirus in the blood of an AIDS patient, even if we select patients with so-called high viral load. I made that statement after having reviewed all the existing literature. I made that statement during the Mbeki conference in Pretoria in 2000, and feel assured that that statement has never been refuted or corrected. So it's a known fact that we cannot demonstrate one single particle of retrovirus in the blood of AIDS patients, even if we select those tagged as presenting with a high viral load. Now, the samples used routinely for the past 10, 15 years to measure the so-called viral load are plasma samples. But feel sure of one point: the technique used to prepare such samples in view of the application of PCR technology to amplify some nucleotide sequences, these samples are prepared by techniques that have absolutely no chance to isolate retroviral particles. We know very well for the past 50, 60 years what are the techniques required for proper isolation of retroviral particles. And I can assure you that the way samples are being prepared from human blood plasma to prepare for PCR technology and demonstration of a so-called viral load, these samples do not contain one single retroviral particles. So, what is being recognized in these samples by the PCR technology? In my analysis of that, what's being recognized are nucleotide sequences trapped by, present in the circulating DNA of these patients, and DNA sequences that derived from the HERV present in all these patients. So the results of so-called viral load determination have strictly nothing to do with a totally hypothetical HIV viremia. Another misleading factor in the analysis of all of this is the claim that there are many mutations of HIV. To understand that, we have to keep in mind that the HERV, human endogenous retrovirus, we have are not a single species. There are many, many families of HERVs, and they do not all have an identical genomic sequence. So it's no surprise whatsoever that identifying these sequences have quite often led to variable results, and which were interpreted as an indication of HIV mutations. This has nothing to do with HIV mutations. It simply indicates that we are recognizing by PCR technology nucleotide sequences originating from a large variety of families of HERVs. A third confusing, possibly misleading fact that has to be faced and analyzed is that in the original publication by Barré-Sinoussi and Montagnier and their group at Pasteur in 1983, there is a most elegant electron microscope demonstration of typical virus, retroviruses budding on the surface of some lymphocytes. But if you carefully analyze that paper, and here I must apologize to emphasize an old paper because I'm pretty sure that if I would ask you who has seen that paper, I would have a very, very limited answer. It's a paper published in Science in 1983, but a paper that did a lot to put scientists on the wrong track because it was relying on molecular markers like reverse transcriptase, which, as we have indicated earlier, is not an acceptable marker, and two, that was consolidated by that famous image representing most typical retroviruses. But the group at Pasteur was using extremely complex cell culture. And if you analyze their paper very carefully, you find out that that picture has nothing to do with an AIDS patient. They mixed in their culture human lymphocytes isolated from circulating blood, cord blood, placenta-derived cord blood lymphocytes. And we have already mentioned that the human placenta is extremely rich in human exogenous retroviruses, and that therefore, in all probability, lymphocytes originating from the placenta carry the same endogenous retroviruses. So, having mixed in their culture lymphocytes of placenta origin, then having presented a picture in which the so-called budding viruses, which were clearly demonstrated, are budding on the surface of cord blood lymphocytes, not on any cells derived directly from the patient, is an incredibly misleading interpretation of a very clean electron microscope picture, but that picture put a lot of people on the wrong track and made a lot of people accepting the basis of the HIV/AIDS theory as reported by the group at Pasteur in 1983. So, to stress the point again, these retroviruses clearly observed in that paper can readily be explained by the mixing in their cell culture of lymphocyte of placenta origin, and most likely have strictly nothing to do with the AIDS patient they were working with at that time. This is that famous picture, but let's not go into details on that, because I don't feel there are many electron microscopists in the room this morning. So, what I was hoping to convince you this morning is that three problems were still of a perplexing nature if we want to consolidate the statement that HIV does not exist. These three problems were the viral load, were the claimed frequency of HIV mutation, and three typical electron microscope images in the historic Pasteur 83 paper. I hope I succeeded in convincing you that in these three cases, the role, the interference of human endogenous retroviruses has been absolutely critical and has provided a scientifically acceptable explanation for these three factors that otherwise could be regarded as misleading. Obviously, the interference of HERV has to be part of any scientific analysis of AIDS research. It cannot be ignored. And if you look in the gigantic HIV/AIDS literature, you probably will never find any reference to them. They are cautiously ignored. In conclusion, I like to stress that I'm certainly not the first one to call the attention to the misleading role of HERV in AIDS research. There is a paper published, I guess, in 2008 by Voiset and others in London, where they speak with considerable detail about what they were describing as the confounding role of HERVs in retroviral research. And I can only confirm that, yes, indeed, there is an enormous confounding role of HERV in HIV research. But if we keep all of this in mind, I feel it's becoming clear that HIV as an exogenous disease-causing microorganism has never been proven to exist, and that therefore spending billions of dollars for HIV research is a total waste that should be stopped. If we had plenty of money, perhaps that would not be so important. But in these dramatic days of a worldwide financial crisis, we cannot afford to waste any research budget. And to give such monumental amounts of funding to research which is very strictly centered on HIV is really not acceptable and should be stopped. I feel that we have a considerable responsibility to indicate that new lines of research should be developed and funded. But for that, we should liberate all the funding wasted currently on HIV/AIDS research. So the concluding point I like to make is that I do not regard the question HIV exists or not as an academic question. It's a lot more important than that. Let me tell you this. Imagine that the funding for AIDS research would be currently evenly distributed. Let's say 20% for retroviral research, 20% for behavior, 20% for malnutrition, 20% for the abuse of drugs. If we would have such an even distribution of research funding, then I would agree with Peter Duisberg and saying that perhaps the question of the existence of HIV or not is rather of an academic nature. But as you know very well, practically 100% of the monumental, the billions of dollars put on AIDS research for many years by now are allocated to research exclusively centered around the HIV hypothesis. So this has to change. This has to change by developing new research programs that would develop the chemical causes of AIDS epidemics, chemical causes that were so well described by Peter Duisberg, Rasnick, and Köhnlein in their 2003 paper in Journal of Bioscience. So those of you who would like to look at that discussion, HIV existing or not, as an academic discussion, keep in mind that it is not academic because from the answer we bring to that question depends the reorientation of AIDS research, which is very urgently needed in view of the total lack of success of all what was based on the HIV hypothesis. For those of you who were interested in my analysis of all of this, I would like to bring to your attention that I've written basically for scientific readers a paper which is currently in press now in the Journal of American Physicians and Surgeons. And it's in press now and should be out very, very shortly. And it contains a very large amount of very precise scientific references that I hope are adequate to bring total support to all what I was trying to tell you this morning. For those of you who would instead like to read more along the same lines, but more for the lay public, I cannot avoid making a little bit a reminder announcement for a book I published with Jean-Claude Rousset already 2-3 years ago, which has been translated into, initially it was in French, but it is translated into English. And you can find on the table at the registration of this meeting a little literature giving you the specifics about that book. So thank you for your attention, and I hope I succeeded in bringing to your mind that there is something about HERV that cannot be ignored in the analysis of HIV/AIDS research. Thank you very much for your attention.